CPMV is the Cowpea Mosaic Virus. When we inject it in the tumor, we wake up the patient's immune system to hone in and attack the tumor. They train other immune cells to then systemically go through the body and kill tumor cells anywhere within the body. Using a plant virus for cancer therapy is not an intuitive idea. It was rejected many times, but eventually I got the right set of data, the right mindset, that I landed the ground finally.
All answers are with black-eyed peas. Can we cure cancer with a virus that infects plants? Can we solve the global food security crisis? And what does it mean to create a sustainable future? I'm excited to be sitting down with Dr. Steinmetz, professor and vice chair of the Department of Nanoengineering at the University of California, San Diego, the founding director of the Center for Nano-Immunoengineering, and co-director of the Center of Engineering in Cancer. Thank you so much, Dr. Steinmetz, for joining me today. I'm a big fan and was really excited about sitting down and chatting with you. As we think of viruses and things that make us sick, so we saw during COVID that, people passed away, and then we also saw that, HPV can cause cancer. I think you actually have a really unique perspective on just viruses in general. Do you mind sharing that with us?
Yeah, so I do think about viruses differently, and my lab works with plant viruses specifically, and yes, plants have viruses too. But I don't look at them as pathogens. I really just look at them as tools, as a platform technology that we can engineer and repurpose to do something good. And this all goes back to my undergraduate studies in Germany. As a young student, I attended the lecture by Uli Kommandeur, and he talked about using plant viruses to produce pharmaceuticals in plants. And to me, that was sort of mind-blowing. Like, we can learn from biology, study biology, and then repurpose that biology to do something good. And in this case, they were using the concept that plant viruses make copies of themselves in plants. So can we repurpose that property to make copies of pharmaceuticals proteins in plants? And this is kind of where my ideas came from. I joined his lab shortly after that lecture. And today, rather than using just elements of plant viruses, we're actually using viruses as novel therapeutics.
And to my understanding, you're doing quite a bit of work with nanotechnology and CPMV, or black-eyed peas. Do you mind telling us a little bit about nanotechnology? What is it? Just for those at home who might not know.
So yes, nanotechnology is the science and engineering of very small matter, specifically the nanoscale. So what is the nanoscale? We go from a meter to a millimeter to a micron down to the nanometer. So it's 10 to the minus 9 meters. Or in other words, if you think about it, say you look at a human hair, that's an object. The width of a human hair is very small, but still visible to the naked eye. A nanoparticle would be 100,000 times smaller than the width of a human hair.
Wow.
So yeah, nanotechnology concerns the science of the very tiny objects. And so what do plant viruses have to do with nanotechnology? Well, essentially a plant virus can be regarded as a naturally occurring nanoparticle. Nanoparticles are nano-sized objects, and plant viruses are nano-sized objects. And we can really define them as a nanocarrier, because naturally it's a protein shell at the nanosize scale that packages a cargo. The cargo is the genome of the virus. So for nanotechnology, we take out that natural cargo and replace it with something useful, say a protein drug, a chemotherapy drug, or some other molecule of interest.
And so how did that evolve in your research to CPMVs?
Yeah, so CPMV is the Cowpea Mosaic Virus. It has its name from the symptoms that it makes in plants. So it infects cowpeas or black-eyed peas, same beans that you can buy in the supermarket. And when it infects the plant, it makes mosaic symptoms on the leaves. So that's why we call it the Cowpea Mosaic Virus. But that's not why I became interested in it. I actually studied the Cowpea Mosaic Virus since my PhD research back at the John Innes Center in England. And there I developed chemistry to start modifying Cowpea Mosaic Virus. So started looking at it as a nanoparticle rather than a plant virus. And I came then to the US, where I did a postdoc with Mary Manchester here in San Diego at the Scripps Research Institute. And there we started using Cowpea Mosaic Virus as an imaging tool. So we modified it with fluorophore so it would light up when injected into a cell or an animal subject, and used it to image disease. When I started my own independent lab, we were more interested in therapeutic applications. And our approach has always been to study the biology and then to learn from the biology how to best repurpose it. And through these studies, what we learned is that Cowpea Mosaic Virus, or CPMV in short, interfaces really well with the immune system. And so even though it's a plant virus and doesn't infect humans or animals, it's recognized by the immune system, and it's very potent. So we started using it as an immunotherapy and vaccine platform.
It's very interesting. And I recently saw your presentation at the Precision Oncology Conference. Very insightful. And you had mentioned you've started a partnership with Spain. And what I found so interesting was you were doing research on canines, but you said canines are not canines, they're patients. And I just loved your perspective of that, given it is, it does have life. Do you mind talking to us a little bit about your research there and what you're doing?
Yeah, very happy to. So we discovered, and this goes back 10 years now, we discovered that when we inject Cowpea Mosaic Virus into a tumor, that it generates a very potent anti-tumor immune response. And I'll explain how this works in a bit more detail before I get to the canine studies that we're doing these days. So about 10 years ago, we noted in drug delivery studies that the control sample, the empty particle, would shrink the tumors. So the initial intention was to develop these plant virus particles for drug delivery. And so our sample had a drug encapsulated, we would deliver it to a tumor, and that shrunk the tumor due to the chemotherapy action that we delivered. But we also noted that the particle itself would have an effect on tumor growth. And we didn't really understand it at the time, but thought we should study this further. I then met an immunologist at a conference, Steve Fiering, whom we collaborate with until this day. And together we probed whether we could use the Cowpea Mosaic Virus as an intratumoral immunotherapy, where the idea is that we take these particles, which we knew are immune modulatory, inject them into a tumor to agonize the patient's own immune system against the tumor. So the idea here is that even though it's a plant virus and it's non-infectious towards mammals, it has all the bells and whistles to be recognized by the immune system. Mm. So when we inject it in the tumor, we wake up the patient's immune system to hone in and attack the tumor. And it's not only attacking the tumor that is being injected, but the immune system trains on these immune cells. We form specialized immune cells that will recognize the signatures of the tumor. They train other immune cells to then systemically go through the body and kill tumor cells anywhere within the body. And so what that means is if you have a patient that has one injectable tumor, but also multiple metastatic sites, we only need to treat the one tumor that maybe is a localized tumor, easy to inject. And the particle will engage with immune cells, wake them up, agonize them. These immune cells kill the primary tumor cells, learn what these look like, and go through the body, everywhere in the body, to kill also distant metastatic sites.
Oh, wow. So does it work with solid tumors and non-solid tumors?
So at the moment, we're focusing on solid tumors.
Okay.
But we've tested in mouse models, we've tested all kinds of tumors, breast cancer, colon cancer, glioma, head and neck cancer. And what we find is that the therapeutic effect is highly potent and tumor and tissue agnostic. So in a way, we think of it as a personalized immunotherapy that does not need to be personalized.
Oh, I like that.
And so we don't really need to understand the tumor. I don't need to know the mutations of the tumor, what type it is. All we need is an injectable tumor. And it's not not a more toxic drug that we developed. It's also not more targeted. It's really just like a flip to switch back on the patient's own immune system to clear the tumors.
Interesting. So would this replace chemotherapy or?
I don't know if it would necessarily replace it. It works in great synergy with chemotherapy and other therapies as well. For example, the chemotherapy helps to debulk the tumor, and then the immunotherapy can help to train the patient's immune system to prevent recurrence. But it certainly, in our studies, we have shown we can reduce the dose of chemotherapy that is needed. We've seen the same with radiation. We can reduce the dose and the treatment frequency. Also with other immunotherapies, the checkpoint inhibitor molecules that work well in some patients, but not in all patients. Our data indicate that our therapy using the Cowpea Mosaic Virus also works in mouse models that are refractory to treatment. So I think from a translational point of view, it has very potent efficacy as a solo agent, but would also greatly synergize with existing therapies.
So is this relatively novel, using plant viruses versus maybe other creative, options available?
Yes, it is a novel approach towards cancer treatment. Mammalian viruses have long been used in medicine. As an example, some of the COVID-19 vaccines were based on mammalian viral vectors. There are other researchers in the world using plant viruses as a technology platform, but to date there's no approved plant virus-based therapeutics. So it's certainly a novel area.
Right. So this could be this could be a trailblazer in the industry.
Yes, I certainly hope that we can move this forward into a phase 1 human trial. And towards translation, we have begun to work with vet clinics. So we started collaboration with several clinics in the U.S. At Dartmouth College, Oklahoma State University, amongst others. And we have a collaboration with 2 universities in Spain. And what I want to stress here is these are vets. We are not using these animals or dogs as a test subject. It's somebody's pet that is diagnosed with cancer. So we are using this sort of 2 ways. One, to de-risk our technology, because many new approaches work well in mice, but then don't translate into human applications. Mm-hmm. So testing our novel therapy in the dogs helps us to get more validation and more proof of concept that it might work in a human. It doesn't prove it will work in a human, but it gives us more confidence. But on the other hand, it's somebody's pet diagnosed with cancer. As of now, it's a compassionate use, so the owners sign up for this treatment as compassionate use. The animals we treated have come in with heavy disease burden, large tumors, metastatic disease, where they have maybe with standard of care 1 to 2 years to live. But in a recent trial in Spain, we showed that many of the dogs we treated lived for another 5 years, tumor-free.
Wow, that's amazing.
Yeah, it's a very, very exciting for us to see these results, and also the vets are very excited about it.
Do you think you could actually leverage this to other diseases potentially outside of or in addition to cancer?
Yes, we're working on applying this to other diseases. In the past, we've also worked in the past and currently, we're working on cardiovascular disease. During the COVID-19 pandemic, we also explored applications for infectious disease. We made a plant virus-based COVID-19 vaccine candidate. We were a little bit late to the game, but we showed it worked, at least in an animal model. So I think there's lots of opportunities and potential for plant virus nanotechnology.
That's very interesting. So what got you into becoming a scientist? You said you grew up in Germany, went to school in London, came to the US. Tell us a little bit about, what stemmed or inspired you to become a scientist.
Yeah, I started in high school. I loved all the STEM fields, in particular biology, and so I started studying biology. And then when it came to my master thesis, right, where you have to pick a specific topic to dive in deeper, I was really thinking hard about what do I really want to do. And it was not until I saw this lecture by Uli Kommandeur, who talked about using biology towards novel materials or novel therapeutics, that got me really excited about engineering applications. And while I'm not formally trained in engineering, I am now a professor of nanoengineering, and applying the concepts from biology, tweaking biology towards new applications.
And from my understanding, you also were a figure skater?
Yes. Yeah, I was in the national team for Germany for roller figure skating.
Okay.
And I won the German championship, was second in Europe, and scored top 10 positions in the world.
That's amazing. I used to be a figure skater when I was a child. Growing up in Canada, skating and hockey was a thing. But it taught me a lot of discipline. What has your journey taught you?
Yeah, it certainly taught me to always stand up and try again until I succeed. Right, learning a new jump is hard work, and it turns out you fall a lot. And the important part is to just always stand up and try again, and sort of fail again, fail better until you succeed. And the roller skating, I think, has prepared me well to be a scientist, because the worlds aren't that different. Right, to learn a new jump in the skating ring, you have to be really focused and always stand up and try again. And I fell a lot, but I stood up and eventually succeeded. And as a professor, writing grants, writing papers, right, I had a fair number of grants rejected, but I always sent them back in. And I am a scientist engineer, right? It's not winning the grant on the first try is fine, right? You can fix the problems that are there, and then try again until you succeed. Yeah, I also try to give my trainees this perspective, right? Be bold and daring. It's okay when you fall. Just make sure you stand up and try again.
Yeah, I saw that as part of your teaching philosophy, which very much resonated with me. And something that you said was how to fail well. How would you describe failing well?
Yeah, I think first off, don't ever take it personally when a grant is rejected, right? It's easy to get upset about it and to feel like, oh, the reviewers didn't understand my point, but it's okay. It's not the reviewer's problem that they didn't understand. It's your problem. You didn't explain it right. And so, whenever a grant review, right, or any rejection comes back, I think to just step away from it for a few days and don't approach it personally. It's not personal. And we're all scientists, engineers, so we can look at this, look at the data. The reviewers gave you comments. Just look at them as data and data points that you can now address and fix and find a solution to come back stronger. And we always say fail again, fail better, and then succeed, because usually it takes, right, when you want to bring across a new innovative idea, I think it takes time to convey that message clearly. And it also takes time maybe for the audience to digest it and really understand it.
Yeah, and I always find, especially, we as in society, we don't talk about our failures enough, right? We talk about all of the things that went well. We talk about our successes. We talk about, the where you've now become versus all of the steps it took to get to that point. Do you feel it's the same as well in your field where, maybe people shy away from talking about their failures, because I love that. I think we need more of that. I think we need to be more open about, because in my personal opinion, failing equals success, right? Because you learn from that. And roller skating, figure skating, you definitely fall a lot. There's a lot of injuries if you don't do it well. Like, what is your take on that?
Yeah, I think we should highlight our failures just as much as the successes. And yeah, because typically we only talk about all our successes, but at least in my lab with my trainees, when I win a new grant, I obviously share the great news with them, but I also tell them how often I had to submit that grant to get it funded. And now my trainees are writing their own grants and fellowships. And I can see, right, the frustration when they get their first grant rejected. And I always go at my folder and show them how many grants I had rejected, but then how many I also got. And it's, right, it's a journey to get there. And it's, I think understanding the process helped me become a better scientist, helped me to, when I write a grant, to very carefully think about what are the reviewers going to say. Right? Try to predict, based on the previous comments, what are they going to say? What are going to be the issues? And it's hard to predict that ahead of time, but at least when I get the reviews, I always read them carefully and don't approach it in a way like, oh, the reviewer is wrong. It doesn't matter if they're wrong or right. This is what they read and understood. And I think, and to drive innovation and creativity, maybe the first idea wasn't the best idea. Maybe it was fair that they rejected this grant, and it made me think so much harder, collect so many more data to prove the point. No, this using a plant virus for cancer therapy, there's not an intuitive idea, and it was rejected many times. But eventually I got the right set of data, the right mindset, and the right message delivered that I landed the grant finally.
Mm-hmm. You definitely have that hustler mentality. That word has come up quite a bit this week. What is, as I was preparing for this conversation, I also found that you created the Nanoman, which I thought was super cool. Can you tell us a little bit, like, what inspired the development of Nanoman?
Yeah, the Nanoman is the tiniest superhero on an important mission. Go, Nanoman. Find that tumor man.
Super awesome.
And yeah, this was a collaboration with a Retro Forward musical writing team, Krista Knight and Barry Bringer, who are based in New York City. We randomly met at a party, started talking about science and the theater world and the skating world and how they kind of all merged together. And we together came up with this idea, like, why don't we make a musical that explains the concept of nanomedicine? And we performed the musical several times, a couple of times in Berkeley and New York City, off Broadway.
That's awesome.
And it was really fun. It was geared towards general audience, typically attended by young professionals. The play was called The Nanoman, Turn Off the Dark. And it was drama and musical theater. We had Dr. X treating a cancer patient. But then she realized the chemotherapy, which was what she used at the time, just went everywhere in the body. And so she invented the Nanoman to carry the chemotherapy, avoiding the healthy cells, so keeping the patient more healthy during treatment, and delivering it to the tumor. And at the time, we then came up with this jump and run, sort of old school video game that Dr. X would control the Nanoman, and the Nanoman could communicate with Dr. X, and it was eaten by macrophages, and it got stuck in some tissues and organs, and Nanoman would always communicate the problems, right? I was just eaten by the immune system. I didn't make it to the tumor. And then Dr. X talked to the students, we need better engineering tools. And so with each iteration or each level, if you will, the Nanoman became better and eventually reached the tumor.
That's amazing. Was this actually, could you actually play the video game? Is this available to
Yeah,
other scientists?
we have a web page, the nanoman.org, and there's sort of an early version of the game, but it's still work in progress.
That's very neat. Very neat. It's it really reflects, sometimes you're in the right place at the right time, or spontaneity and like-minded people coming together to really provide those opportunities, because I think, you're starting to see quite a bit of movement in the industry and in the space, and to make it fun just because it's heavy, and to create a video game in theater is just a great way to educate others as well, because I think there's potentially a lack of literacy when it comes to science. And I think, we saw quite a bit of maybe a breakdown in trust over the last few years. What do you believe? What do you not believe? We're inundated with a ton of information. So how do you decipher that in a way that makes sense, especially when it comes to treatment plans or clinical trials or a variety of different options available to patients? So that's really interesting. Is there a potential opportunity to commercialize this and make it available to young kids?
I think there would be a pathway to do this. I think it would require maybe more collaboration, or maybe people watching this might become interested to team up or have new ideas. The current version of the game is sort of very old school, jump and run, like a Super Mario game, 2D, not 3D.
Nice.
But I think, there's a potential to make games to educate kids about the human body, right? It's interesting to look inside, and I think this could be very interesting games to be on a mission, like to deliver a cancer drug, or to be on a mission to deliver a drug to the brain and other organs.
Mm-hmm. Which might also inspire the future leaders of tomorrow.
Yes.
Especially in the field of STEM. As you're using your current technology, it could also be used in other facets like food security. So the Sustainable Development Goals #2 really targets global poverty, food security. And I think based on the research you're doing and some of the novel technology, how can that actually be used to create a sustainable environment and maybe help tackle our food security issue?
Yeah, so a few years ago, we started applying the concept of nanomedicine to agriculture. And we're approaching this by using plant virus nanotechnology. And right, you might ask, so why use a plant virus to increase plant health? But plant viruses evolved to get around plants and get around soil in ways that synthetic materials have not. And to maybe explain how we got into the thinking of using plant viruses for agriculture, came from my research in the medical field. Now, if we look at how doctors have used chemotherapy for many years, is you use a dose that can kill as many cancer cells as possible while avoiding killing the patient. Now we use nanotechnology that can interface better with the human body and maybe target that chemotherapy to specific cells, the tumor specifically. So we can use the same thinking in agriculture. Today we use chemicals, pesticides, and flush the field to kill as many pathogens and bugs as possible without killing the environment. But what if we could package these pesticides into nanoparticles and deliver them to the site of infection, and thereby reducing the dose that is exposed to the environment? And one particular problem that we became interested in are nematodes. These are roundworms that are feeding on the roots of plants. And they cause massive losses in the US and worldwide every year, and they're very difficult to treat. And the current approach is to essentially flush the field with nematicides, chemicals or pesticides. But the particular challenge is that these worms feed on the roots of plants. So they have to traffic through the soil down to the root level. And many of these pesticides don't have good soil mobility. And based on research articles, it seems that more than 90% of the pesticides applied end up in groundwater. Because they have to apply a very high dose, kind of flush it down to get it to the root level. And this exposes an enormous risk to the environment.
And humans
And
potentially, if they access. Yeah.
yes, humans and livestock. And so we thought, well, why not use a plant virus to deliver this pesticide? And right, the idea here is, again, we learn from biology, repurpose biology, and interestingly, plant viruses naturally interact with nematodes, with these roundworms. And it's in fact the roundworms that traffic the plant virus from one plant to another.
Interesting.
And so we thought that means, number one, the plant virus can reach the root level. And to find these nematodes, so what we could do is we take out the natural cargo, which is the genome, and that makes the plant virus infectious. So by removing it, we also make it non-infectious towards the plant. And then we load the particle up with the pesticide. So we can apply it on the field. It will diffuse down to the root level. The nematode will engage with it to traffic it to the next plant, but in the process, we kill the nematode because the plant virus was loaded up with the pesticide. So in laboratory tests, we've now shown this works. It seems to be a simple idea, but those are usually the best ideas. And we are now working with the USDA, who's setting up sort of an indoor field test on tomato plants that are infested with nematodes to see if it works in an agricultural setting.
Interesting. So is it a spray or do you put it in the crop?
Yeah,
what
so
is it?
That's a really good question. So in the lab setting, we've applied it by just pipetting it on the top of the soil and then add water to mimic rain or watering the plants, and it will diffuse down to the root level. That might not be how we use it in field. There's various drip systems or even systems that are feeding nutrients and water directly to the roots. And that's how we're currently testing it together with the USDA.
So you're doing some really interesting research with the UC San Diego Materials Research Science and Engineering Center with a focus in engineering living materials. What is engineering living materials?
Yeah, so engineered living materials can be two things. In one definition, it's a material that incorporates biological matter. And to give you an idea what that might entail, within our center, the MRSAC, we are working on sustainable materials and materials that are more environmentally friendly. So there's a team of researchers that developed plastics, bioplastics, that contain microbes. And when these plastics find their end use and are left in the environment, the microbes become activated and destroy the polymer. So there's no residual plastic left in the environment. In other areas, it's to use materials to enhance the properties of biological materials. So in one application that I'm working on with a team of scientists, we are working at the plant-human interface.
Okay.
So what is the plant-human interface? So one of the goals is, can we enhance plants with synthetic parts so that they communicate with farmers, right? The idea being a baby, when it needs something, it cries and lets the parents know, I need something. Plants don't communicate, and oftentimes farmers find out too late that something was wrong, that there was an abiotic stress, right, a drought or lack of minerals, nutrients, or some pathogen attacking the plants. And often they find out too late, and it's too late to really do anything about it, and the yields are lost. So the idea is to have a sentinel plant in the field that could sense these abiotic or biotic stresses and send a signal to the farmer. On the other hand, another idea is that can we enhance the plant, say with a hydrogel, to change the morphology of the plant. And to give you one specific example that we're working on, we together with a material scientist, a chemist, and a researcher with expertise in soft robotics, we put a hydrogel under the leaf of a plant, and upon light stimulus, we can change the morphology of that hydrogel, resulting in closing the leaf. And so why is that useful? Now, when plants go to sleep at night, they actually close up their leaves, and they do that to conserve water. So avoid having water evaporate from the leaf and store the water. So if we would realize there's a drought coming, and this would empower the farmer to close the leaves, even during daytime maybe, to conserve more water, maybe reducing the yield but not losing the yield. And these are some really big ideas that we proposed a few years ago. But we are now making sort of in nano steps. We're moving forward, realizing these ideas. So in the lab setting, we already have a plant that has a hydrogel incorporated, and upon electrical stimuli, we can close up the leaves.
That's super cool. So I think you were telling me there's like a chip that you would put in your hand. You'd close your hand, and then you would automatically see that leaf of that plant close as well.
Yeah. So currently it's still a wired solution, but we're on next iteration hopefully will be wireless. So we're sending an electrical signal by just pushing the skin, sending an electrical signal to the plant that will then close up.
And if this were to work in a large landscape, this could actually make a huge impact on just agriculture in general.
I would say we're still at early stages developing the technology. This all started when we moved to UCSD and 2018, meeting a whole new group of researchers and really developing this as a totally novel idea and research direction. And I think I'm also really grateful that we were awarded this big center grant, the MRSEC, or Material Science Research and Engineering Center, which is a really, really unique platform to bring together scientists with one common goal, but with different expertise, right? Plant sciences, biology, material scientists, soft robotics, to come up with these cutting-edge ideas and realizing them. We are really at the early stages in developing the concepts and technology. But if successful, I think it could have a huge impact.
Well, I'm excited to kind of continue to follow your journey and see where it goes. STEM is a growing field, and with every career, I think you overcome a variety of different challenges. You're a trailblazer in your field. Have there been challenges that you have overcome that, without those challenges would have been like lessons learned? If you could go back and maybe change a couple of things, would you do anything differently?
I'm not sure I would necessarily go back and do things differently. I think the process of having certain failures is required to grow and to really learn and become who you are. So yes, after the fact, you always know more than you knew before. But I don't think going back and just doing it better right away would get me to where I am today. I think it's the failures are really part of the journey and learning from them. And I think for young scientists entering the field, I think what's really important is to find something you're interested in. To me, it was this idea of repurposing biology, using plant viruses. And for everybody, there will be something different. But it's trying to find that thing that you're really passionate about. Because in order to be a successful leader in the field, you need time, dedication, focus, and that's easier to achieve if you find something you're excited to think about, you're excited to work on. And so, my advice is find something that you can really be passionate about, but then invest time, focus, and drive to come up with innovative ideas. And then the thing is, once you get to that point and you have something that's truly unique and different, people will say no, right? That's not the standard norm. And I remember as an assistant professor writing grants to say, I'm going to use plant viruses for medical applications. Initially, I was told no many, many times. And even within my department, a faculty member suggested, well, the plant virus approach is kind of so unique. Why don't you switch and use a lipid nanoparticle, something that's more accepted in the field? But I didn't do that because I felt like no, that's not going to be me. I'm the one that develops plant virus particles. So I stayed true to me and my ideas, and eventually succeeded. And yeah, today I have many grants funded, and drive this successful research lab.
Well, I mean, the Steinmetz Lab is very well recognized across the globe. You've over 200 published articles, newspapers, chapters, and that's due to, of course, your success and your hard work. Thank you so much, Dr. Steinmetz. This was a very enlightening and insightful conversation. I'm going to look at plant viruses very differently and black-eyed peas than I probably ever would have. I appreciate you being very open and sharing your story with us, and really look forward to continuing to watch your journey and seeing how things unfold. So thank you so much.
Yeah, thank you so much for having me.